I wanted to specifically thank our advocacy partners for being here. Jacob's Heart, Unravel Pediatric Cancer, Team G Foundation, Michelle, who doesn't belong to any of them, and everybody else, because it is so important for us to have your support, and it's because of you that we're here, we're where we are. So we are here today because kids do also get cancer, and in fact, it's not as rare as we may think. In total, there are 43 children in the US that get diagnosed with cancer, and it is a classroom full of children. The average age of diagnosis is 6 years old, and it comes out to be 1 in 300 kids get cancer by the time they turn 20. Despite the progress that has happened in this field, cancer is still the #1 cause of death by disease among children. And few people know that some childhood cancer diagnoses are actually terminal. And Libby will talk more about what that means to a parent. But we do have some diseases in childhood cancer space, like DIPG, or diffuse intrinsic pontine glioma, which are terminal at diagnosis. So that means these kids don't have a fighting chance from the start. They just don't have any effective treatments at all. And then the other thing that few people know is that because of the way that we treat childhood cancers today, which is with very aggressive chemotherapies, very aggressive radiation, bone marrow transplant, so once a cancer comes back after that, it is likely incurable. So in total, in California alone, we have 500 children a year that are out of treatment options for their cancer, either at diagnosis or at the time of cancer recurrence or relapse. So before I talk about Treehouse, I wanted to share a little bit about how I got to this field. So initially I wanted to go into medical school, and I started as an undergraduate at the University of Toronto, and I got a summer job at the hospital for sick children. And so I wanted to be an oncologist, but then I realized, working on neuroblastoma, which is a type of pediatric cancer, I realized that oncologists are often very limited in terms of what they can do, because some of these diagnoses are so devastating and there is no treatment option, and all they can offer sometimes is words of comfort to their patients. And so I realized, or I thought, that it's only through research that we can really change the story in a big way, and we can change those odds. So I went on to do my PhD in neuroblastoma in Vancouver at the British Columbia Cancer Agency. And there I learned about bioinformatics, and that was the time when sequencing technologies that David was talking about kind of came of age. And everybody was thinking that bioinformatics is the field that's going to come up with cures, that's going to really change the story. And so because of the great reputation of this group is really the top bioinformatic group in the world, that I came to Santa Cruz, and I'm very excited to be able to leverage all these amazing resources and amazing expertise that David has talked about to really change the story for pediatric cancers. And in my journey in this field, I have really learned firsthand the importance of community involvement and the importance of patient advocacy. And so shown here, this is Jordan. Jordan is one of the very few survivors that I know about, long-term survivors of high-risk neuroblastoma, which is one of the most devastating diagnoses one can have. And so in my PhD, we ran into a situation where we were in the middle of grant funds, and so we basically ran out of money, and we were thinking about switching directions because we just didn't have money for this work. And then Jordan's father called us, and I remember that phone call forever. And he had this tremendous survivor guilt because his son survived when others didn't. And so he talked to us, and he said, "Please continue doing what you're doing, because I don't want Jordan to be the only one." And so we did, and that work led to the identification of a new drug target for relapsed neuroblastoma called aurora kinase B. So I'm going to switch gears a little bit and now talk about the science behind Treehouse. So David gave a great introduction into cancer genomics, and he talked about how DNA, which is letters, which is basically composed of 4 letters A, T, C, G. And so that provides a set of instructions for how cells and the whole body functions. And then when you mutate the DNA, or what you get is really spelling mistakes in these letters throughout life, then eventually you get cancer, which is abnormal cell growth. And so with these new technologies that David talked about, we can now read these letters. So we can read the DNA, and we can specifically read DNA of cancer cells, and we can try to understand what those spelling mistakes really are. And so this approach has been very fruitful for adult cancers. And the way that we do this is we read the DNA of adult cancer cells, and then what we do, we look for common spelling mistakes. So we look for spelling mistakes that are common, for example, for all breast cancer, or for many breast cancer patients. And then a pharma company would learn about that and make a drug that would then help thousands of those patients. But of course, the problem is, while we can do the same thing for pediatrics, we have a very big problem. So in pediatrics, it turns out that there is much fewer mutations than we see in adults. And because our whole field depends on mutation-based drug design, this is a problem. There is also fewer cases than adults. And probably the biggest problem is that recurrent or common mutations are rare. And what this means is that really for every case, you need to develop a unique therapy, which is not how the pharma companies work today. So this is a big problem. And we really, to address pediatric cancers, we really need a paradigm shift in the way that we interpret genomic information. So David talked about data sharing and the importance of data sharing and consolidating genomic information. And I think that is the solution, that is the paradigm shift that we're looking for. And so I'm going to walk you through what we do once we get those data, when we have the data from many, many different cancer patients. And so specifically in Santa Cruz, we have accumulated data from over 10,000 cancer patients, both adult and pediatric patients, that we have access to. And so we have this tool called Tumor Map, developed by George Stewart's group, and Yulia Newton is a graduate student working on this. So this tool allows us to spot similarities between different tumors. The idea here, the importance of bringing in adult cancer data, is because adult space is where the drug development takes place. As I mentioned, adult cancers such as breast cancer, prostate cancer, they're common, and so pharma companies are developing drugs for those conditions. And so what we can do with Tumor Map is we can identify situations where a rare child's tumor or a rare tumor is similar to another tumor type where there is a drug available. And so what you're seeing here, and you have a handout on Tumor Map that you can take a look at. So these dots, each dot represent an individual cancer patient or an individual cancer patient's genome. And so these dots are laid out in this two-dimensional space according to the similarity of genomic profiles, so the DNA similarity between these different tumors. And then in this particular view, these dots are color-coded by the type of cancer that these patients have. So for example, here we have breast cancers, they're all in blue, and then here in yellow we have brain tumors, we have gliomas. And so what you can see that for the most part, the same color clusters together. So all the breasts are clustering together. But you also see situations like this where you see the wrong color infiltrating this green blob. And this is very significant. This actually means that even though this could be a bladder cancer, for example, but at the level of DNA, it is more similar to another tumor type. And so maybe it should be treated as that tumor type. And we all know examples of family members of friends where two people have seemingly the same diagnosis, one person responds and the other person does not respond to the same treatment. And we think this is why, because at the molecular level, those tumors are actually different and they should be treated differently. And so now I would like to share with you our first patient, our first pediatric cancer patient that kind of got us started on this journey of California kids cancer comparison and kind of taught us how we can use the tumor map approach, or what we call pan-cancer analysis approach, to help individual children with cancer. So this patient was an 8-year-old boy, had a very rare sarcoma, was treated at the British Columbia Children's Hospital in Canada. And so initially was treated with a standard therapy, which is very aggressive chemotherapy and radiation, as is typical for these children. And then 2 years later, the tumor came back in his lungs. And then genome sequencing was done to read the spelling mistakes that are found in this tumor. And this particular mutation was found. And as I mentioned, we are finding these mutations in children, but this is a rare mutation and there is no drug, and maybe only very few sarcomas have that mutation, so there is no pharma company working on that specific drug, and so even though we know what the mutation is, it's back to square one for that patient. So what do we do? And so what we did is we built a tumor map where we placed this patient's tumor genome in the context of all the data we had here in Santa Cruz. And again, so individual dots here represent patients, and then they're color-coded by the type of tumor that they have. And so what you can see, what you can appreciate here, this is a sarcoma, and so we have another cluster here of sarcomas. And so what we maybe or may have been expecting is that this patient would cluster with the sarcomas because it is a sarcoma. But what we saw is that actually at the level of the DNA, this patient's tumor was similar to another type of tumor called neuroblastoma, which happened to be the tumor I was working with in my graduate work. And so I'm not going to bore you with the details of what we did next, but we kind of dug deeper to understand the pathway or what's happening at the molecular level, why this tumor might be why these cells might be growing uncontrollably in this patient. And so this is what we have come up with. So this is a normal cell here, and you can see all these squares. They're individual genes. And so what we think is that these genes, or what we know, is that these genes normally function in a controlled manner, and that they can lead to controlled cell growth. But what we're seeing in our patient, in patient 1, is that all these genes are red, which means that there is too much of the gene product, of the product of those genes. And so it's not the mutations or the spelling mistakes we're looking at here, it's really the amount of the gene product, which is not normally looked at in these genomic studies because it is impossible to determine what is too much without comparing to something else. And because we're able to compare, we can say, okay, this patient has too much of this gene, because we have tens of thousands of other tumors to compare it to. And so what we have thought first is that, so this gene, ULK, is actually a very, very well-studied gene in neuroblastoma. And remember that this patient actually clustered with a neuroblastoma in the tumor map. And so when we communicated this information to the treating oncologist, he decided to actually try ULK inhibitor, so a drug that can get rid of this ULK, and so to do this. And so he did that. The patient had a partial response where some of the metastases reduced, but then others grew. And from looking at this, you can appreciate why, because there is these two other genes that are driving the tumor growth. And so we looked at this case more in detail, and then we realized that actually there is an inhibitor of this molecule called JAK1, which is downstream, and maybe by getting rid of this, we could do something better. And in fact, there is a JAK inhibitor that was available, and the clinician decided to try that next. And so what we saw with that is that the patient had a very good response to the JAK inhibitor, and he had a turnaround in his disease. He was well again, he was playing, he was running around, which was great. We were very happy and very hopeful about that. Now, unfortunately, eventually the tumor did come back, And unfortunately, the patient did pass away from his disease. But what we learned is that this approach can provide new hope and can actually extend life for these patients where seemingly there is nothing else that could be done. So by calculation of the treating oncologist, he thought that we bought 2 years with this molecular work for this patient. And in fact, we are very honored to be part of patient one's journey. He actually was aware that he was involved in this groundbreaking work. He was very excited and he was very proud about being part of this work. And in fact, one of his dying wishes was to, instead of using patient one, he wanted us to use his real name. And so hopefully next time we gather here, I would be able to tell you his real name. We're working out the logistics of it, but that was his wish. And because of him, we started California Kids Cancer Comparison, which is one of the demonstration projects of the California Initiative to Advance Precision Medicine. And as part of this initiative, we are doing the same thing that we did for patient one, for 150 kids with difficult-to-treat cancers in California. And we're working with 3 hospitals, Stanford, UC San Francisco, and Children's Hospital of Orange County. And so the best analogy for what we're doing that I have is molecular detective work. So we're given the most difficult to treat cases, the most hopeless cases where there is no treatment options remaining. And then what we do is we look at the genomic data and we compare that patient to tens of thousands of other patients we have in our database. And our goal is to come up with hope for every single one. So we actually don't stop until we come up with hope. Now, it doesn't mean that we're curing or we are coming up with cures, but at least what we want to do is we want to come up with something else that a family can do as opposed to nothing. And so I'm very honored to report that in 12 cases that we have done since April of 2016, when we started doing this prospectively, for every single case, we were able to find leads. So we were able to use this approach to come up with new potential treatment options. And so just very briefly, I will tell you about one of our new cases that we just finished working. It's a case, it's a patient treated at Stanford, a 2-year-old with stage 4 hepatoblastoma, which is a liver cancer. And so just like many of these cases, the patient went through a lot, so chemo protocols, surgeries, and then out of treatment options. And so Foundation Medicine, which is a common genetic test that's now done, was done and again revealed this mutation. So we are finding mutations, but the problem is there is no strong drug that can really fix this. And so yes, we know that there is a mutation, but we can't really do anything about it. And this particular test was actually developed for adults too, so they're looking at a very limited set of genes. And so when we did the tumor map analysis, we learned that this patient here, so the patient is here marked with this Google Map icon, and it actually clustered, so this is a zoom in of this placement. It clustered with hepatocellular carcinoma, which is an adult liver cancer. And it was actually very unexpected that this very different pediatric liver cancer would cluster with adult hepatocellular carcinoma. And so it turned out that adult hepatocellular carcinoma there are 4 potential treatment options that we could come up with based on the similarity to hepatocellular carcinoma, and they're listed here. So we presented this to the oncologist, and he decided that he would go with pazopanib, which is an aurora kinase inhibitor. And the reason he chose that one is because it turns out that there will be a trial opening at Stanford imminently of this drug, and so this patient will be candidate, and so it's much easier for the family to be remain at Stanford, as this trial is happening locally. And so from what we know is that the patient is going on this trial, and we're hoping that this will work for him. So our big vision is that not just for the 150 patients, but really for every child with cancer, no matter how difficult or how simple their diagnosis may seem, but we want to sequence everyone's genome and analyze the genome at diagnosis. And we really want to compare every child's genome to a network, to a global network of other data, so that we can have all of the available information from the beginning and also access to all available treatment options suggested by such analyses. And we believe that really to accomplish this, we need everybody, all of us working together. We need companies, we need patient advocacy group, we need clinicians, researchers, individual community members working together, because this is really a community problem. And I think together we can really make this a reality. So I want to thank first and foremost patients and families. So those dots in the tumor map, those 10,000, more than 10,000 dots, those represent patients, cancer patients that have donated their data, or they have agreed for their data to be part of these research projects. And if we didn't have those dots in the map, we would have nothing to compare these individual children to. So this is really important, and we really, really want to acknowledge the trust and the support of individual patients. And then also our partners, UCSF, Children's Hospital Stanford, CHOC Children's Hospital, BC Children's Hospital in Vancouver, our advocacy partners, Jacob's Heart, Unravel Pediatric Cancer, Alex's Lemonade Stand Foundation, Kids vs. Cancer, Team G Foundation, and the funding also from the state of California. So these are just some of the children I have met myself throughout my journey in this field, and these children continue to inspire me. Unfortunately, many of them have passed away. So thank you.